Well, good evening, everyone. I'm Matthew Scott. I'm president of Carnegie Science, and I'm as excited as you are about tonight's speakers and tonight's subject. We're going to go on this incredible journey together. One of the things I wanted to comment on, I'm relatively new at this, 6 months now, and one of the things that's been incredibly gratifying is the kind of loyalty and level of interest of our community in these talks and in science in general. That's not a typical thing. Hundreds and hundreds of people come out on a stormy night to hear about science, and we've had great audiences time and again for these lectures. We're actually doubling the number of these lectures for next year because they're so, all right. And it's because of you, so thank you very much. So I have a few comments then by way of introduction to tonight's thing. So in 2012, not long ago, a Dutch entrepreneur formed a nonprofit organization with the goal of establishing a permanent human colony on Mars. It's called Mars One, and the plan is to send a robotic mission to Mars in 2018, followed in 2025 by the first crewed spaceship carrying 4 individuals. The ride will take 7 months. There will be no return ticket, and every step of the journey will be documented in a reality TV show. Given our perpetual fascination with Mars, and tonight exemplifies that, and of some people with reality TV, it might not be surprising that over 200,000 people nominated themselves to go on this non-return journey. Now, I happen to know about a separate competition in which people could nominate 7 people they wanted to send to Mars on a no-return basis, and that had 4.3 million entrants. Now, we might think of these Dutch plans as mostly a publicity scheme, but Mars One claims it is serious. Elon Musk, the founder of SpaceX, who last year announced his own plans to colonize Mars, is certainly serious. And then there is Dennis Tito, the California financier, who is planning a mission to send 2 people on a trip around the red planet as early as 2018. Some scientists are actually supportive of these private missions, or at least they recognize their possibilities. According to a NASA website, a crewed mission to Mars is currently at the edge of technological feasibility. You'd like to know where exactly on that edge. As of last December, NASA anticipated sending its own manned spacecraft to Mars in 2035, so sign up. If humans ever do get to Mars, they will know exactly what to expect thanks to the dozens of flyby, orbiter, lander, and rover missions that have been sent to the planet since the 1960s. These missions have helped us understand how Mars formed and how it evolved. They have taught us that Mars is home both to the solar system's highest mountain and its deepest, longest valley. And they have shown us that its surface is rocky, cold, and sterile, and that dining options are limited. One of those missions, the one we will hear about mostly tonight, has also taught us that the surface of Mars was once very different. And this is where it gets so intriguing. It had an environment habitable for life as we know it. NASA's Mars Science Laboratory mission, which monitors the Curiosity rover currently on Mars's surface, is the largest and most ambitious mission ever flown to the red planet. Tonight, we are honored to host 2 people knowledgeable about its goals and discoveries, Marc Kaufman and Andrew Steele. Our planned 3rd speaker, Pam Conrad, had a last-minute family event that requires her attendance, and she apologizes for not being able to be with us. I can promise you we will bring her back at another time. Each of our speakers will speak for about half the time, 20-25 minutes each, and then we'll have a question and answer session with discussion with the audience, and that's what these microphones will be for. So please plan some questions as you're listening to the talks. So first up, to give us an overview of the Curiosity mission is Marc Kaufman. Mr. Kaufman is a science writer and editor for the Washington Post who has covered NASA and space since 2005. His most recent book, Mars Up Close, Inside the Curiosity Mission, was published last year to wide acclaim, and I think some of you may be lucky enough to get his signature on it on a copy of the book tonight after the talks. A 1973 graduate of Columbia Graduate School of Journalism, Mr. Kaufman has had an illustrious career that has taken him all over the world, as I was just hearing a little bit in talking with him, including India, where he lived for 4 years, and Afghanistan, where he reported on post 9/11 events. He has written articles for numerous magazines and websites, and his many awards include the Eugene Meyer Award from the Post for war zone coverage of Afghanistan. His 2011 book, First Contact, Scientific Breakthroughs and the Hunt for Life Beyond Earth, was included on the Kirkus list of best nonfiction books of 2011. And the wonders of this, of course, are that it could almost qualify as a fictional book too, and yet it's true. So it's really wonderful. Our next guest, the second guest, will be our own Andrew Steele, who's a staff member at the Carnegie Geophysical Laboratory here in Washington. And he will tell us about efforts to identify signatures of life in the Gale Crater. He's a microbiologist by training. So what is he doing playing around up there? And as he says, an astrobiologist by choice. So that's the answer. He works to develop protocols, instrumentation, and procedures that aid in the detection of organic matter in samples from the early Earth and elsewhere in the solar system. He is a member of the Sample Analysis at Mars team, SAM, which is responsible for the instrumentation aboard Curiosity that has searched for and found compounds of carbon on Mars's surface. He, like several geophysical laboratory scientists, is an active member of the NASA Astrobiology Institute, and he is the science leader and principal investigator of a NASA astrobiology technology grant responsible for testing instrumentation for solar system missions. So he goes to places that are on Earth but very harsh, and this work has taken him to the Arctic every summer for the past 8 years. The late astronaut Sally Ride, who served for several years as a Carnegie trustee, once said, quote, "Studying whether there's life on Mars or studying how the universe began, there's something magical about pushing back the frontiers of knowledge," unquote. Tonight we will hear about a magical journey that not only pushes back frontiers, but is a landmark in our efforts to answer one of our greatest mysteries, whether life might exist or once have existed in a place beyond Earth. So, ladies and gentlemen, please welcome Marc Kaufman and Andrew Steele.
Hello, everyone, and thank you so much for coming out here on a night like this. It looks like Snowball Washington is about to return, and hopefully you'll all get home before then. And unfortunately, it's really unfortunate that Pam won't be here. She's a force of nature, a wonderful speaker, incredibly intelligent. She started out as an opera singer, turned into a scientist. And in most of her emails that she sends to me, she signs off as Warrior Princess. So unfortunately, she's not here, but for the next time you'll know to come listen to the Warrior Princess. All right, here is the star of our show, Curiosity. The not quite, little rover that could, because it's a little bit too complicated for that, but it has been on Mars for 2.5 years and has found remarkable things. But first, as we say in journalism, in newspapers, a headshot of our subject, which is Mars. And of all the planets, I think it would be fair to say that Mars is the one that humans relate to the most. It's because it's red, because it's been identified so much, and there have been so many stories told about it. Going back to the god Mars, and later all the canals that were identified, although they didn't exist. And then later, this is one of my favorite stories. Before the Viking mission, there was a great deal of enthusiasm and feeling that they were going to find life. And the esteemed Carl Sagan was one of the people most involved in the astrobiology there. And one of his colleagues at the time told me just recently that Carl was not only utterly convinced that there was going to be life found, but that this other gentleman was in a room when Sagan learned that some lights that were going to be on the Viking capsule had been descoped, so they weren't going to be on at night. And apparently Sagan just smashed his hand down and said, "Okay, we're not going to see the creatures that come at night. It's on you." So Viking lands, which is, an absolutely amazing feat technologically, but this is what they find. It's really not that great. I mean, it's there are very few features. This was in the in the northern plains, as we'll later find out. That's not where stuff really happens that much. But there was an effort to determine whether or not there was life. And it became extraordinarily controversial, still is to this day. There are still people who argue about whether one of the tests did show life, but for a whole variety of reasons, it became clear that while the US and NASA were capable of sending missions to Mars and landing them there, they were not capable of determining whether or not there was life. They were just way too far ahead. So the combination of the mixed and confusing results and what you see here resulted in a hiatus of almost 20 years, where the idea of life and Mars kind of bifurcated, and there were very few missions, if any, if I remember correctly, and it was somewhat written off. However, in the last 15 years or so, because of some of the orbiting equipment, both cameras and other instruments, they've been able to determine that there used to be a lot of channels on Mars. There used to be deltas. There used to be river basins. And they certainly didn't see that initially in Viking, although they knew then that there were some deep water channels. But this is the kind of stuff that is a lot more interesting to people who are familiar with these things. Plus, they found different kinds of minerals, minerals that can only be formed in water. In this picture, we have some sulfates and also some clays. And suddenly Mars was starting to look a lot more interesting. It had this history of water. And once there's water, then who knows what else there's going to be. So curiosity happens. It is by far the largest vehicle ever sent by humans to another planet, the most complicated also. Landed 2.5 years ago now to enormous acclaim and started its mission. But as we'll see, seldom do you see. So I had the good fortune to be out at JPL on the night of the landing. It's a little scary to see that many ecstatic engineers. I have to tell you, it was like a surreal experience, but it was also just magical, as JPL is generally. And this is where they landed. Gale Crater, 91 miles across. Where they landed is roughly over here. And the plan was to go down here and then look at these incredible striated rocks, which was the basic reason why Gale Crater was chosen, because those kinds of rocks can tell all kinds of interesting stories. So they land pretty close to where they wanted to land, and there you go. As you'll see, here's Gale. And we'll come back to this later, but all of this is blue because it's low. It's 1 to 3 kilometers below everything else here, except for this big Hellas Basin here. And that is, as we'll discuss later, of great significance. And Gale is fairly close to that. This is called the dichotomy. It's where Mars kind of splits. All right, here's have you noticed that there are a lot of selfies that, this is a somewhat vain kind of rover, but it's also extraordinarily capable. It has 2 chemistry labs. It has the mass It has cameras with precisions and also with zoom that they've never had before, and it has a drill on its long arm, which is the first to go to Mars. It's unlike Viking, which had the mission of trying to determine whether or not there was life on Mars. The goal was here intentionally more limited. Was this a habitat? Were there habitats here that could support life? And again, the logic is, okay, from Viking we don't know how to determine yet whether or not there is life. I think Steele will have more to say about that, but because there's been progress. But the idea is, could life have existed here? And remember that first picture of Viking, from Viking? It's like, very uninteresting, no features. Look at this. I mean, this is the kind of stuff that makes geologists just dream. This is wonderful. And the plan is, to go up here and to look around. They determined from orbit that there were large deposits of clay and of sulfates and of hematite, also all of which are formed in the presence of water. They need water. They have to have it. And so the plan is to go to an entry point and then climb up partway and determine a lot of the history of Mars and also whether or not those habitats could have supported life. One of the first things they discovered, and I guess they probably already knew it, but Mars really isn't red. The red planet is red just a little bit, kind of like, the blush of rouge. It's iron oxide, but that's what most of Mars is like, or a lot more of Mars. And this is, I find this particularly interesting because it's one of the first ways in which we're disabused of our misconceptions about the place, that it's a lot different than we think it's going to be. Excuse me? This was the landing site, and early on, as in within the first week or two, there was kind of a fateful decision that had to be made. The plan, as I said before, was to go down, I think it's 7 or 8 kilometers, and then go up Mount Sharp. But they found as they were coming down, and they already had a sense of this, that there was a really interesting area here where 3 different rock formations come together. And as geologists will explain, that's indicative of something really interesting. And so after a certain amount of both risky and scientifically savvy back and forth, the team decided that they would go to this area, which was the opposite direction. And everything according to these missions generally is pretty set out. But here they decided, okay, this is formally a mission of discovery. We're going to go down here, and it's a good thing they did, because what they found was the most interesting place, I believe, that has ever been visited on Mars by far. Look at this. I mean, they never ever have seen this kind of formation of rock, and it turns out that some of this is mudstone. So if this looks like a dried out mudflat, it's because it is. It just got dried out 3.5 billion years ago, but it's still basically the same thing. This was not from orbit. They didn't know that there were also a lot of clays here because they were covered with that red dust. But it turned out that they had a lot of those minerals that everyone looks for in terms of potential water, and the presence of water in potential habitable areas. Just to go back for a sec, this is the arm of and the drill is on here. It's 7 feet long. As you can imagine, it's kind of like pretty cumbersome. It goes like this. It's very hard to move around. Some people have criticized Curiosity for not doing more drilling. It's just really, really difficult, really slow. This gives an insight into how and why. But here was the first drill that was done, the first full drill at this area, Yellowknife Bay, into a mudstone, and what they came up with was just mind-boggling. By the way, this is, they say, the drill hole the size of a Sharpie. Just imagine, how precise that, or the resolution of the camera. But this is calcium sulfate here. They found 20% clay in the soil. You'll see the soil also is not red. And the story that this told them was that this area had water. They didn't yet know for how long, but it had water for a period of time that created this mudstone, and that the water had a neutral pH, so it was, in the words of John Grotzinger, the project scientist, then it was drinkable. You could sit there and, if you were there and still alive, you could drink it. And then the calcium sulfate would have come in later, after this other had been formed. So this was as Grotzinger would say, a real home run, because at the first real serious drill site, they found what was clearly a habitable area. It had water, it had an energy source, it turns out to have had all of the elements that were needed for life, the phosphates and phosphorus and sulfur and hydrogen and oxygen and so on. And so within the first 4 months or so, or 5 months, they had this phenomenal result. Nonetheless, and this, in my National Geographic book, we of course have to do images that describe what things might have looked like. This is what our artist rendering is of part of that area, or some of that area, and it was done with some scientists kind of signing off, saying that this was at that early stage of their knowledge of Gale Crater, what things might have looked like to some extent. That there was an alluvial fan coming down, and then there was this water of some sort that might have been deep, might have been shallow, they didn't know at that point. The plan was to be there for, I don't know, a couple weeks. A year later, they leave. Partially because things are hard to do there, but also because they were finding so many remarkable things. And they go on this finally this trek down to where they're going to go up the mountain. And here are just some of the things that they find that seem intriguing to me. An iron meteorite. This is the kind of thing that you could find on Earth as well, but you're just driving along, you see one of these things. This is it's called a yardang, which is a rock that's carved by the winds up there that are very strong. Here's crystal that they found as well, and here's a dinosaur bone this actually is a picture from Mars, but that is not a dinosaur bone. And Steele will get more into this, but during this period, there also is years of work go into determining whether or not there are organic materials up on Mars, and there are a lot of confounding issues involved. But finally, during that time, they do determine that there was a particular organic that, as you can see here, shows up in one of the drill holes, but not the others. And since you have a control, then you could say this is for real. And this was the first time that you could say for sure that there were organic materials, the building blocks of life, on Mars. Also, as they're driving down, they find these kinds of what they interpret as being the remains of deltas all along this route. This would be the kind of the final end of it. I think these are sandstones rather than mudstones like we had in Yellowknife, but it's the same basic idea that this is a signature of water, of lots of water. And this, I guess this was just the end of last year, is what the project scientists and the team conclude was the case at Gale. That not only was there like water and mudstones and all that shallow stuff, but that there was like a big lake. And you'll notice that there wasn't a mountain in the middle of it at that point, but that came later. But a finding that there was a significant lake and that it lasted for I think millions to tens of millions of years is what Grotzinger said. And he's a very conservative kind of scientist. Many are, but he in particular, and that he was part of this effort or this decision, to me is kind of remarkable because it changes the understanding of Mars. There were theories, hypotheses saying that there might have been things like this, but to kind of ground truth it, to find geologically and geochemically the signifiers of that, remarkable. A little bit before that, and this is circling around to some news that actually is going to come out tomorrow. And because it's embargoed, I can't tell you about it, but I'll hint at it broadly. In those northern plains, this is a paper that came out in 2013 finding, this is Earth, this is what a delta looks like, this is Mars, and these are what are called inverted channels. They're the middle part of rivers that used to be, and then they become fossilized, and the lighter things blow away, and what you're left with is the pattern of what was clearly a delta. And in a manner that I don't at all understand, they were able to determine that this delta at the end made a deep dive. So in other words, it was going into a deep body of water. And this is some of the news that will come out tomorrow, which is part of a theory that's been around for a long time, that that the northern part of Mars, which is much lower, that maybe there was a northern ocean there. And tomorrow, if you were to Google, or to look on some newspaper websites about a northern ocean, you might see a story even by me saying that there was a kind of we've hit a real milestone. Because in the world of astrobiology, of habitability, I mean, what could be more conducive to life than to have a large ocean? Now, it's not a slam dunk. We don't know for sure yet that this is the case, but we have two local scientists, very senior people, who did work that I can't say exactly what they found, but what they found is really, really interesting, and it involves the Northern Ocean. And more about this tomorrow. Also, in late last year, backtrack. Two scientists, also local people in the area, had a science paper in 2009 describing plumes of methane, the gas methane, an organic gas, that they said were coming out at different times and in different places on Mars. It was kind of controversial because it was so paradigm shifting. If it turned out to be true, then it was really important because it meant that these organics were somehow underground and were coming up. This can be done in a variety of ways. It could be done through various changes in rock and whatever, but it could also be done biologically. And in fact, on Earth, 95% of the methane is created by living organisms. Doesn't mean that that's the case on Mars, but we now have we have water, we have lots of water, we have methane, and we have a road ahead which involves going up these remarkable mesas and looking and digging in and analyzing what's in these different layers, and hopefully finding signs that not only was this habitable, but sometime in the future that it was inhabited. Thank you very much.
Hello. So, I think, yeah, there we go. Can you hear me? No, not at the moment. I'm afraid my dancing skills are not what they were. I just have to wait for my talk. So I was supposed to kind of sandwich in between Marc and Pam Conrad. And Pam was going to talk a lot more about the habitability issues that Marc alluded to. I was going to interject between the two with the subject that I am most passionate about, which is how to find life on Mars. It's a very, obviously, a very interesting scientific question to look at as far as we're concerned. I like this picture of Curiosity mainly because this was before landing, and that was my field area in the Arctic that they put up there, and that's green. That's a delta that has some grass on it. So from my perspective as a biologist, I've had to learn an awful lot about geology, and you'll see as my talk goes on, there's a reason for that. Now, why are we interested in Mars? Why do we think that Mars might have developed life? Well, about 4 billion years ago, the thought is that Earth and Mars are really quite similar. Today, obviously, the story is a lot different from the perspective of Earth and Mars being similar to each other. But at one point in time, round about here, Earth and Mars were quite similar. Now, if you view this graph as a 12-hour clock, you can see that this bit here, life, we think, and it's open to debate, at about 7 minutes past, we got simple unicellular organisms. And really nothing anything else until about 25 where the more complex unicellular organisms started, then multicellularity. Your dinosaurs are here for reference, and just about here, about 5 or 6 seconds to midnight, we appear. And we're trying to make sense of all of this as far as our own planet goes. But back here, Earth and Mars are really quite similar. So if there was life on Earth, could life have formed on Mars? Now, this is a real eyesore of a graph. I showed this at a talk recently, and one of the postdocs said they thought I was showing a map of the Tokyo Underground. These are all the different metabolic pathways that life has derived over time. Each one of these little red marks or blue marks is an enzyme, okay? So looking at that, there's your classic biology, your TCA cycle is in here. So when you look at it in that respect, biology is a massive building project. Each one of these enzymes is just like a huge machine, okay? Depending on how they work, they just kind of bully the substrate around, really. They're all kind of mechanical in their functions. But this derived from very, very simple beginnings. Now, this is the bit that we're really, really missing. The rock record on Earth is really sparse back at the time when Earth and Mars were very similar. Two of the Mars meteorites, we have date from that time. There's some of the earliest planetary material we have, apart from a few zircons and some rocks in Greenland and South Africa and Canada. What interests me most is this transition here between abiotic chemistry, the chemistry that is done in meteorites, the chemistry it's done, it's simple organic chemistry, to what becomes prebiotic chemistry, a chemistry that has the potential to lead towards life, and then becomes biochemistry. That's the transition that really interests me and what I work on. But the search for life on Mars is more than just looking for an answer, does Mars have life or not? Yes or no? Well, in the Viking era, as Marc alluded to, the no answer that came up from the mission itself sort of put a stop to Mars research for about 30 years. They were very disappointed. Carl Sagan was very disappointed that nobody visited Viking at night, or during the day, or at any other time. So that really kind of stopped things for a while. But no is not a negative answer. If there is no life, why isn't there life? If Earth and Mars were very similar at that point in time, why didn't Mars develop life? And that tells us about Earth. Why is Earth different if Earth did develop life? Why? So the single most compelling argument for Mars exploration is a scientific search for our own origins. It's not just to say, are we alone? But it's also, where do we come from? Now, one of the ways in which I study, and so I can see several of my colleagues in here who also study them, is we've had a way to study these for a while in meteorites that come from Mars. So this rock was quite happily minding its own business, sat on the surface of Mars. When another rock comes in, hits it and blasts it from the surface of Mars, it then goes around the solar system for a while and crashes into Earth. We generally find these in Antarctica or in deserts. And we know they're from Mars because during this event, most of these are made of silicate minerals, which when they bubble are like glass. They trap the atmosphere, and when you release that atmosphere, it's very similar to what we found with Viking. Without Viking, we wouldn't know that these came from Mars. So when you look closely at these, as a gentleman called Dave McKay did in 1996, who published a paper on relic biogenic activity in one of these meteorites, basically saying there was evidence of early life on Mars. And that's, as a microbiologist, that's where I came in. I actually got a postdoc with Dave McKay as a microbiologist to look at this meteorite a bit further. He had 4 lines of evidence. These kind of golden rosettes in the meteorite were made of a carbon mineral called carbonate. At the edges, there was iron sulfides and magnetite, which is an iron oxide, which shows a disequilibrium process that may be due to life. And then there were all these purported microfossils that could be evidence of past life. Now, I'm a biologist, and I approach things from the point of view of a thing called a null hypothesis. So if I want to find something, I automatically assume the opposite and try and disprove that hypothesis. It's just a way I was taught as a biologist to do this. So the first thing that I did was get a sample from Dave, and he said, "What's this material?" And after looking at it for a while, amidst the furor of all the investigations going on this meteorite, it was basically obvious that this was a terrestrial microbe. Yet all the techniques looking at this meteorite to search for life were saying there was no life in the meteorite, but there was Earth life. And that was kind of can't see the wood for the trees scenario a little bit. This is slightly complicated, but this is basically carbon. This is graphite that is trapped in those carbonate globules. This is a result of my research. And from looking at this association of this kind of carbon, which is just like lead in your pencil, this kind of stuff, which we know can form from life's remains, but also can form from a chemical process where you can take carbon dioxide and iron oxides, and basically as you cool a liquid containing those, you will make graphite with magnetite in this system, okay? So what I'm saying is there's a very simple chemical explanation for most of the observations in that. At that point, I had to get another job. So looking even more closely at the meteorites, in a lot of these meteorites, in some of the minerals, you find these tiny little inclusions. Now I have some instrumentation that I use that allows me to interrogate these while they're still trapped below the surface of the mineral that I'm looking at. This is a very, very thin wafer of sample with the little inclusion trapped in between it. And when I look very carefully at that, I find carbon associated with these minerals. This is pyroxene, this is spinel, it's surrounded by olivine. This is an organic carbon residue. Very interesting, apart from the fact that this mineral assemblage cooled down from a magma. Now, we can think of all sorts of scenarios where life could survive on Earth. We see it in boiling hot pH 1 liquid on Earth. We see it 8, 9, 10 miles up in the atmosphere. But right slap bang in the middle of a magma is not somewhere that you'd normally associate with biological activity. I see it in a whole host of different meteorites spanning the entire age range of Mars. So what's happening here, and this is a model, we can't prove this, but this is the most likely scenario, is that the fact that the carbon can be dissolved in these silicates as a magma, and as the magma solidifies and crystallizes, the carbon has no oxygen to react with. If there is, it comes out as CO2 in your normal volcanic eruptions, lots of CO2. But in Mars, there's not that much oxygen in the mantle to react with as Earth. It comes out as soot. This is basically, unfortunately, not life. And then Mars was very, very kind to us in 2011. Of all the meteorites we looked at, there's only a subset. There's over 100 Martian meteorites been identified. There's only a subset that have been seen to fall. So the Earth tends to try and make soil out of any rock, whether it be from Mars or the early solar system. The organisms get in it, water gets in it, it weathers, they try to make rock. But this meteorite from Mars was seen to fall, picked up within 4 weeks. And off it came to my laboratory, as well as others. So what we see here is the surface of one of these polished samples. And then if I focus down, this is in reflected light, so the light's bouncing off this. This is the light's going through it. And so I can focus down into the sample, and I see all these nice little blebs, which are very similar to these kind of microfossil size, bacterial size, etc. And they do contain a lot of carbon. So many investigations later, lots of different techniques thrown at this. And this is the advantage of having the sample here. This scale bar here is about 200th the width of a human hair. This tiny little mineral assemblage that was trapped in there has been cut out and put into a transmission electron microscope. And you can see, when I look really carefully, all the organics that I'm seeing is associated here in this area 4. This is associated with a mineral called magnetite, which was the same mineral I found in Alan Hill's 84001, associated with the graphite, and with the other meteorites associated with the igneous carbon. But the exciting thing about this, and this is still ongoing work, is it also contains, as well as organic carbon, it contains organic nitrogen. And this is a spectrum of just that little spot there. That's 50 nanometers, roughly speaking. And we contain all these nice organic functional groups. So something's going on here that is different to the other carbon that I showed from the igneous processes. And what I think is happening, I mean, if you review the evidence, it's tiny, it's got carbon in it, there's a lot of organics, there's a lot of functionality, the carbon isotopes are light. It's very compelling, but what I think is happening is on Earth, for many years we've known about these hydrothermal vents at the ocean floor. And during the release of volcanic gases and fluids in this system alongside similar minerals that I see in that meteorite, you get organics produced. That's why one of the reasons why you have such a wonderful ecosystem around these things. The really odd thing about this, or coincidence about this, is these were discovered the same year Viking landed. We know so much more about these than we do of this. And these tiny little mineral grain, that tiny little mineral grain in that meteorite, I hypothesize that is a teeny tiny little black smoker. Unfortunately, no tube worms or crabs or shrimp or bacteria yet. So meteorites are only a piece of this puzzle, okay? With all the faith in the world, that meteorite, when it came in, it hit this sandy desert with a lot of carbonate. Then it hit, a big plume of dust went up, and the dust slowly rains down, and the meteorite is cracked. So all those little cracks become just an entranceway into the center, and it's a grain sorting mechanism. The smaller the grain, the deeper it gets, okay? So I can't, in all faith, separate that from some of my observations. I can look at this material that's really trapped in these inclusions, but is that the most interesting stuff? No, because if there's organisms there, they're going to be growing on those cracks, and that's where I want to look. But I can't do that because it's contaminated. I can be smart about how I look at it, but I have to be conservative, right? I have to be able to falsify my null hypothesis. And to do that, I have to make sure that I'm not just looking at something either I put there that the instrument puts there or the Earth put there as I'm making my measurements. So they are contaminated by the terrestrial environment, even though the Tacint meteorite is very minimally contaminated. You lose all context. We don't know where on Mars this came from. We have no idea. It's just a random chunk of rock that just happened to be unlucky enough to be next to an impact and hit Earth. And that's why we need the robotic in situ analysis that Marc has shown with this fabulously engineered machine that we have. The next step, and I'm just back from a trip to the Jet Propulsion Laboratory, where we're planning the first step of Mars sample return in 2020, and hopefully at some point, human exploration, whether they wanted to go there or not. During all of this, you'll see that when I do this organic analysis, I need to know the context of those measurements. I need to know the mineralogy around it. Why? Because you can make fuel oil from just passing CO2 over catalysts. This has been making fuel oil since the late 1800s, okay? A process called Fischer-Tropsch synthesis. There are all sorts of ways of doing simple chemistry that can mimic life. But that complexity of life itself leaves a different signal. But teasing that signal out is the most important thing. And one of the things you need to do, for instance, I find organic carbon in the middle of an igneous rock that came from a magma. I need to know if that rock is igneous. Is how and why Curiosity is set up the way it is. So Curiosity has eyes, just like a geologist. It had a hand lens imager, just like a geologist. It can rove around the place like most geologists. It's able to do chemistry at a distance. It's got a laser and can just peck at spots and do the chemistry of that spot. And that gives you the elemental ratios, how much calcium, aluminium, silicon in that rock. So what it's able to do is stand off and look. So if you go into that beautiful rock field that Marc showed the Vikings saw, you'll see rocks of all shapes and sizes, but mostly the same color. So you want to be able to know which one of those rocks is special. So you can stand off, you take your image, you look and see the context, and then you can use your laser just to go, oh yeah, that's made of leather. That's made of leather. I'll move on to this one. Oh, this is made of something else, right? And that's how you make your decision to go to a certain place or not go to a certain place. Then you get closer, and it's literally the first thing a geologist will do is pick up a rock. The next thing they do is usually taste it, which is really quite odd. Geologists, please, if you're doing life detection experiments, don't taste your rocks. It has the capability to do contact science. It can image it and get contact, much better understanding of the mineralogy of the sample based on a suite of instruments based on the arm here, including it could drill, it could scoop, it can brush, it can sieve. And all of this is part of sample preparation. The next thing a geologist does is take that rock back to the lab and look at it with more complex instrumentation. But by the time it's got to the geologist lab, they've discounted thousands and thousands of rocks, right? So you look at a lot, you can do a little bit of chemistry on a lot, you can do contact science on a lot less. And in the back of this, there's two instruments to do definitive mineralogy and organic chemistry analysis. Those only get the samples that we think, oh yeah, this is really exciting. There's a really small subset of that. And the decision tree to get there is not an easy decision tree at all to make. I'm honored to be part of the SAM team, which is the Sample Analysis at Mars team. This is about the size of an old-fashioned milk crate, and has several different components to it. This is a gas chromatograph, there's a mass spectrometer, there's a tunable laser spectrometer. This is what is finding methane, and this is what is doing the organic analysis. You can see the different columns in here. It's literally hundreds of miles of tubing and wires, and it's by far the most complex instrument, I think, that it's ever been flown, and it's amazing. It's a real technological miracle that works. And kudos to the Goddard team who put that together. And if you want to know what the sample data looks like, and this is coming out in a paper from Caroline Frissonnet and Danny Glavine and part of those papers, we see a slew of different chemicals, but what you have is a series of wiggly lines. Scientists love series of wiggly lines. And you'd see on the graph that Marc showed before where the chlorobenzene is up above the background, there's a lot of background. We have to contend with an awful lot of background when we're crunching this data set, which is normal. Every instrument does have a background. But you can see we can pick out, there's trichloromethane, dichloromethane, chloromethane, and various other, there's the chlorobenzene coming out. And we can measure all of these, which is a real technical tour de force. Really, it's an incredibly exciting thing. Every time the data comes back, we're all sat there with our computers waiting for that series of squiggly lines and wondering if that series of squiggly lines is going to tell us something different from the last series of squiggly lines, right? And it's amazingly exciting to sit there and wait for that data to come down and crunch that data set. So far, there's a lot of activities that are going on with data that we can't talk about, but it comes out in time. We have to be incredibly careful. It's really robust science and a really robust way of doing science on Mars, but it's also time-consuming and very, very intensive activity to come to these results. But basically what this is showing is confirming a lot of the meteorite work. Now, that Mars does have organic chemistry. It does do organic chemistry. Now, to what extent did it do organic chemistry and whether it transition from the abiotic to biotic state is still in debate. Now, going back to Viking, Gale Levin with the label release experiment claimed that label release experiment was a positive for life. One of the main arguments against Gale Levin was the lack of organics in that sample. But we now know that those Viking measurements might have found some organics, these simple chloromethanes. Whether Gale is right or wrong is open to debate. But what that was is a data point. And as a scientific community, we look at data points and then we review that data with the next set of data points and the next set of data points and the next set of data points. Life detection isn't about a single measurement and never will be unless something with 6 legs walks up and says hello. We're not going to find it unless we do a lot of analysis and a lot of robust analysis. So through my work, I've found all sorts of different ways of making organic carbon and where carbon is on Mars. And that work, we find new forms of organic carbon. The methane story is an extremely interesting story. Yes, it's biogenic. There are non-biological ways to look at it. I can't falsify my null hypothesis on any of this work, unfortunately. But it is something that helps us, to robustly look at the data set. And come up with an idea of how to do life detection, which is the punchline of my talk, I suppose. This is a really complex series of squiggly lines that basically is a mass spectrum of the Murchison meteorite. The Murchison meteorite is a carbonaceous chondrite filled with organic chemistry, revolutionized our understanding of the solar system and solar system processes when it fell back in, I think it was 69. And you can see here, this is the mass in hundreds of units. Carbon weighs 12, hydrogen 1, oxygen 16. And you can see the slew of organic carbons in this, and there's thousands of them. If you look at the way amino acids are formed, you'll see that the meteorite itself contains every possible combination of amino acids. 3 carbons, 4 carbons, 5 carbons, every possible way of putting those together. And the L versus D forms are equal, where life prefers the L form, almost exclusively. There are exceptions to that. So what does that tell us, and what does the research on Mars tell us? Well, for me, the best way, I think, you can do life detection on Mars with no assumptions on life chemistry, right? So one of my favorite films is Aliens. An alien, an insectoid creature with sulfuric acid for blood. Fantastic. How did it do that, right? We have no idea what an alien biochemistry would look like. We have no idea of the morphology. We have no idea what it looks like, what it eats. Nothing. What music it prefers? Nothing at all. We have no idea if it's going to be similar in any way, shape, or form to our preconceived notions of what terrestrial life is. I mean, we can't even define life on Earth. You get 4 biologists in a room and ask them to define life, and you get 27 answers. So, to have no assumptions on life chemistry, you need to make only 3 assumptions with this hypothesis that I generated. And that is that it's carbon-based, and it does not use all the molecules available to it. So of all those hundreds of different types of amino acids in the Murchison meteorite, life chooses roughly about 30, roughly speaking. Of all the possible nucleotide purines and pyrimidines, it chooses 4, 5, a few modified ones here and there. So it chooses only a very, very finite subset of those molecules and concentrates them up. So if you, in DNA and RNA, have A, C, T, and G, you might have in your analysis LMNOP, a different alphabet. But the thing is that those will be concentrated above and beyond what you would expect from organic chemistry. And that's what these observations on Mars are really important, and meteorites and the work I'm doing, is that it sets a baseline, an abiotic baseline. And then you look to see how that baseline is perturbed or changed or different out of what you would expect from the context it's in as far as the mineralogy goes. And that's basically why I do this work, why it's so stimulating intellectually, is coming up with that and then trying to measure it. Now, curiosity will get us some way down that road. It will measure organic carbon, and now we have to figure out what happens. And methane is a great example of that. Is it biologically produced or is it non-biologically produced? But just like any scientific journey, it is just that, a journey. The next set of missions will be informed by this mission. The next set of analysis informed by the data that you get from the analysis that you have just done. That is the scientific method. For me, if Mars has no life, why doesn't it have life? What does that say about Earth? It's a test tube hypothesis. Did Mars get 5 test tubes down the 10,000 test tubes you need to get to life? That's the question. Are there other places that would do that? And the follow-on from MSL is another MSL type rover, which will fly in 2020, because no matter how fabulous our robotic exploration is, and Curiosity is an unbelievable technical tour de force. It's the most complex spaceship we've ever flown. It's run by the most dedicated scientists I've ever worked with, and it's coming up with the most fabulous results that we've had on Mars. But also we need the samples back. I need to be able to look at the scales and prepare the samples at the scales that I have shown you in the data sets this evening. And that, hopefully, the first step of that will launch in 2020. And there will, a rover very similar to MSL will core and cache samples ready for return to Earth. And this is before maybe the smaller people in this audience may get the chance to go and actually explore Mars and look for those signs of where we come from. Thank you very much.
Okay, now it's your turn. If there are any Martians here, we'd like you to speak last to give the terrestrials a chance to speculate. You can come to either microphone and ask in any language you wish. Don't be shy.
Did he work? I feel like Baldur. He could.
Hi. I don't know if you guys are the right people to ask this question, but I'm just wondering if, contamination is such an issue. How do we prevent us from contaminating Mars with Curiosity? And how would you, if you're going to bring samples back, how do you still not contaminate them when you get them?
So the greatest job title in the world is the Planetary Protection Officer. NASA has a planetary protection officer. The slogan is all the planets, all the time. And their job is basically to make sure that we minimally impact Mars from an environmental standpoint. We don't want to contaminate what we're going to look at initially. So every mission gets graded for its planetary protection potential. Viking was baked, I think, for about 36 hours at 120 degrees centigrade to kill as much terrestrial organisms on there as possible. Now, it's not always been the case that has happened, and meteorites can go from Earth to Mars. But as far as a mission standpoint goes, every mission is accredited on its planetary protection potential, and every mission has to go through a series of tests and they grow organisms out of them. They have a whole test sequence that they have to go through. Now, bringing samples back is a really interesting one, because how do we avoid the whole, basically War of the Worlds scenario? Are we going to bring something back that then infects the planet? So there's a whole series of a thing called the draft test protocol to actually address those issues and to sterilize the container and what's called break the chain of possible contamination. Now, it's really interesting for return samples. You would handle Ebola in what is called a class 4 facility for microbiology. This is class 5, because it turns out that class 4 are quite dirty, and you still have to handle the samples. So they have to be cleaner than the cleanest category 4 facility that we have for handling the most deadly organisms on Earth. And that's all put in place by your planetary protection officer, who's Cassie Conley at NASA, and she's got the greatest job in the world.
I would just add to that, there's a real irony and complexity associated with the planetary protection, because specifically because of contamination, we cannot land a rover in an area that might have life, or that might have water on the surface in some way. In other words, some of the places that are most attractive are off the table because we don't want to contaminate them. And however much we would decontaminate before going, it could never be entirely done. And as it turns out, Viking was a lot better in terms of getting rid of contamination than anything that followed. And part of the reason is what has followed is a lot more complex, and that if you put Curiosity in an oven to bake for 36 hours, there would be no more Curiosity.
Mm-hmm.
Hi, my question is about what happened to a lot of the water that they theorize was on Mars. So you talked about a lot of the lakes that might have existed. What's the theory as to where all that water went?
The theory is generally that it just flew off the planet for a variety of different reasons and in a variety of different ways. There are poles the North and South Pole, that have still some water ice in them, and that is perceived as being, I think, about 20 or 30% of what might have been on Mars at one point. But also the water goes into the making of some of those minerals, and there may be large aquifers that are liquid beneath, but an awful lot of it, much of it, just flew away, because what they have found is that the heavy water is still there to some extent on the ground. The light water is not there, and that means it disappeared.
Part of it, if you go to the top of Mount Everest and try to have a good cup of tea, the tea is lukewarm because of the atmospheric pressure. Now, the atmospheric pressure on Mars is about 1% of Earth's. So water goes straight from ice to vapor, and it can then escape through what's called hydrodynamic release. So it gets kind of ablated away from Mars. So the atmosphere isn't thick enough to hold liquid water.
4 billion years ago, was there more exchange of materials between Earth and Mars than there is today?
To speculate, Pre that, you had the period of late heavy bombardment, where a lot of meteorite activity and bigger meteorites than we see today was impacting both planets. So potentially, yes. Although I'm no expert on that particular area, but potentially, yeah.
You folks may be familiar with the concept of panspermia, which is a theory that suggests that that meteorites from Mars or wherever might have been kicked up and flew to Earth, and this would be in the early days when Mars potentially was more habitable than Earth, and the possibility is there that organisms of some sort could have survived that trip, which means, of course, that, and this explains a lot, that we may be Martians.
Actually, to follow up on that, two-part question. Why are we looking for carbon-based life on Mars? And the second part, if we wanted to look for non-carbon-based life, how would we even ask those questions?
Oh, cheers. Carbon-based life, because carbon chemistry is so diverse. It's what we know. And we know that we have an n=1 of what life is like. Carbon, its ability to make four bonds and bond with itself and bond with nitrogen and oxygen, etc., to make the compounds that it needs to react together. It has to be reactive, but not too reactive. It has to like water, but not too much. There's a whole slew of, and it polymerizes in wonderful ways, and you have to get nitrogen in that system, carbon-nitrogen bonds, to form some of the. So it can make bonds with itself, but not too much, because they have to be broken. If you think of DNA and the way DNA zips together, or proteins and the way the proteins zip together, it's much easier to do that with carbon. The next element that I've seen speculated about is silicon, because silicon can do a lot of the same things, but the reaction system is a lot slower. The heat, the temperatures used to drive it is a lot higher. Kind of notion of having a solute in that system is, it could be sulfuric acid, etc. It becomes so alien so quickly that I don't think we'd even know how to begin to detect it on Earth, never mind anywhere else. The other thing about silicon-based life is obviously artificial intelligence and the Turing test, and that has been speculated about, but from a chemical point of view, it's much more difficult to do with anything other than carbon. We think. Maybe we're missing something. It's true. I mean, if you look at the way that we look for the diversity of life on Earth, a lot of it is to do with DNA and DNA sequencing. We know so much of the DNA in the environment, but we don't know very much of the organisms that produce that DNA. We can culture less than 1% of the microorganisms in the environment, but we have the DNA. We know they're from the DNA. What happens if it's not DNA-based? All our assays are completely skewed to unknowable life. So maybe we don't even know the depths of those kinds of lives on Earth. But the search for life on Mars is making us ask those kind of questions.
Good evening, Professor Steele, and thank you for your talk. It was very interesting. Two-part life-based question. You mentioned in your talk, you used the expression, the complexities of life, or I think you used that term. I was wondering if you could expand on what you meant by the complexities of life. And secondly, if you could give us your personal definition of what would be, how you would define life versus inanimate non-life.
That's a good one. Yeah, thanks for that. Definition of life is very difficult. I mean, I'm no philosopher. I kind of have the philosophy that I don't know what it is, but I know how to find it. Really. I mean, it obviously has to replicate. Obviously, is out of balance, out of equilibrium with its environment. It needs to bring in nutrients. It needs to move around. It changes its environment, etc., etc., etc. But a very wise man called Hat Yoda, who used to be a staff member at the lab, who's passed away now, used to enjoy asking that same question of all the biologists that came in. And I sat there having, tried to answer that question and seen many others try to answer that question and flounder. And I'm floundering now. Basically, it has to be able to preserve a flow of information that ensures that it replicates itself perfectly across several generations. And in so doing, it impacts its environment and competes for those resources to outcompete anything else that could interfere with that. Now, minerals sort of do that in the way that they grow, but it's a diffusion and a physical-based process. Life tends to go against those kind of physical processes and burns energy to get more energy. It speculates to accumulate. And that's the one property that I think is the most lifelike, if you wish. And I forgot the first part of your question. Well, yeah, I mean, DNA. I mean, just look at any kind of DNA replication of a gene and a genome. It doesn't just, the word there's an A, it shoves a T. It turns it on when it needs it. So the gene is quiet until it's needed. And then some stimulus comes along and it turns it on. And it makes this massive, great protein. It's almost like going on a war footing and making a tank factory. And it knows how to do that. The external stimulus push it towards that, and it does it. It executes it. And then it controls it. It turns it on. It turns it off. It makes more of it if it's needed. It breaks down what isn't needed. And that kind of complexity, that feedback loop, really distinguishes it from organic chemistry. And then when you look at the complexity of these proteins, I mean, there are hundreds of thousands of amino acids that are built, and somehow those amino acids are coded for by the DNA. So not only do they have form and function, but that form and function is coded for, and then they make the DNA. How did it do that? That's really, really odd. I think one of the most fascinating examples is that somebody went over and took all the chemicals needed to make the genome of a virus. So, your adenine, your cytosine, etc. And then all the amino acids needed to make the proteins in the virus. Put them together in a test tube, and the virus made itself and became infective. And we can't even agree that a virus is life or not. Then what is it? And that hint of complexity, where you'd have to have an organic molecule kind of make a small petroleum factory and then make itself. That's the kind of thing that is that transition. How does it do that?
Just a short additional thought on complexity. We tend to think of life on Earth in complex terms because that's how we live. But as the kind of time chart that Steele showed before suggests and explains, for about 3 billion years on Earth, all of the life that was here was single cell, which I guess would be considered not complex. So it gives a real interesting insight into what we might be looking for elsewhere, because it's all just a question of time. And if you hit it at the right time, then a planet might be doing one thing. If you hit it at a different time, it would be very different. But in any case, to get complex life is extraordinarily hard.
But I've been saying that even a bacteria, it's so complex. That system is so unbelievably complex. And then you put us on top of that, where each of the different cells, instead of trying to kill each other, somehow cooperate into skin and organs and cells and brain and nerves, and then that all comes together. And then somehow that interacts with the environment to change the environment. I mean, the orders of complexity just keep ramping.
I didn't mean to put down single cell open.
Hi. In one of the recent lectures, I heard about if the life on Earth becomes extinct from a cataclysmic event, still there is a possibility of regeneration from a soup of amino acids and the life forming in the atmosphere or much further out. Is there any such scenario in the Mars where you said the Curiosity has found some organic materials? There may be a beginning of that, or the future possibilities for the life there?
I think Steele, you want to.
there's nothing to say that life isn't being made now, new forms of life or new types of life. On the probability curve, you could say if it happened once, didn't it happen many times here? Sterilizing events through impact, etc., tend to, it's like the etch-a-sketch, start again, isn't it? And rub it out, and off you go. But those kind of sterilization events don't tend to sterilize the whole planet. Now, the search for life on Mars is one where we're very much concentrating on the higher probability of there being past life on Mars, not present life on Mars. But we can't really rule out present life on Mars. So at some point in time, if life did evolve on Mars, the cataclysmic event that turned Mars from being, a watery world into a dust bowl, that event may have completely sterilized the planet. And at that point, we may be looking for signs in the oldest rocks of that life. But then, if you want life to start again, you have to recreate those conditions, whatever they were, that Mars had initially. Now, I personally think on Earth that plate tectonics has a lot to do with this, because if you're doing any chemistry, you need a Bunsen burner, and you need energy in that system, and a lot of it, and for a long period of time to get that complexity. So if Mars had that way back then, it seems to not have had it again. So any kind of sterilizing event probably sterilized it for good.
There also are some Mars scientists who are of the view that it is possible that there is microbial life deep underground, that perhaps it was on the surface at one point, terrible things happened, and then it went further down. And that's why some of the methane work is so fascinating, because while the methane certainly could be abiotic, non-biological, it's also possible that this is a reservoir created by methanogens, microbes that create methane, or conceivably even still do. It'll be a long, long time before we can dig down that far to tell, but there certainly are scientists who have that view and are trying to figure out ways to either look at it in a sophisticated way.
Since there is water ice at the poles, it would seem that they might be very interesting targets for exploration. Are they out of the question, either because of the contamination issue or other issues?
Well, I think they would be out for the contamination, I think, if I'm not mistaken, it would just also be really, really hard to land on it. I'm not sure that you could do that. The Phoenix lander was pretty far north, but they didn't get that high up. But yes, I think from planetary protection perspective, it would probably be off the table. What do you think?
Well, one of the interesting things about the poles is Mars' obliquity has changed in short timescales. So those poles weren't always where those poles are now. So as the planet flips, the obliquity changes cause the poles to move around. So they're not that permanent to have had something for ancient life. But Mark's answer is quite correct about potential for current life. But getting there, landing there, doing your investigations on something that's shifting so much. I don't know if you've seen the pictures of the geysers, the CO2 geysers coming out from Mars. Well, have a look at that. And this terrain is incredibly active, and shoving a spacecraft down on that terrain is difficult in the first place. And then to keep it there for any length of time on an operational standpoint to do those investigations is also extremely challenging.
Great. Well, we want everyone to travel safely. I want to mention again that Marc will be signing some books outside, I believe, after this. So that's quite wonderful. And thanks again to our two fantastic speakers this evening.